Hello students. Welcome back to the channel. So in the previous lecture we have studied about male and female reproductive system. In this lecture we will study about gametogenesis. So let's start. Gametogenesis is the process by which gametes are formed. The process of formation of human male gamete, that is sperm, is called as spermatogenesis, and the process of formation of human female gamete is called as oogenesis. Let's discuss about spermatogenesis first. It occurs inside the testis. But for understanding what happens inside testis you need to understand two processes. You might be knowing about these two processes. The first process is meiosis. It is the process in which a cell divides into two, and the chromosomes are divided equally between them. It is divided into two phases, namely, first, meiosis phase 1. Second, meiosis phase 2. The second process is mitosis. It is the process in which a single cell divides into two but the number of chromosomes remains same. Now let's begin the explanation. This is the diagram of formation of sperm inside the testis. This is the initial stage of sperm with 46 chromosomes. Here it is called as spermatogonia. In the next phase it is called as primary spermatocyte. Meiosis phase 1 occurs here. And after it a single spermatocyte divides into two and the number of chromosomes reduced to half. And they are called as secondary spermatocyte. This time the spermatocytes undergo the process of meiosis phase 2 and called as spermatid. And lastly these spermatids develop into sperm. But these sperms are immature. So to attain maturity they enter into retestis. After retestis they enter into the epididymis to gain nutrition. Here you can see the diagram of a human male sperm. This is the head region. At the top of the head region acrosome is present. It helps to penetrate the wall of egg. At the center of the head region nucleus is present which carries genetic information. This is called mid-region or neck. It contains mitochondria which provides energy to the sperm. This is the tail. It directs the sperm to the egg. After penetrating the wall of egg the head region detaches from body, which means only head region enters inside the egg. Now let's understand gametogenesis into a human female. A human female produces egg by the process of oogenesis. Before starting the explanation let me tell you one important point. For females the process of gametogenesis takes place at two stages. Firstly at the fetal stage and later at puberty. Now let's start the explanation. This is the initial stage of egg. Here it is called oogonia. It further develops into primary oocyte. Both these processes take place at fetal stage. It means that the number of egg to be produced by a human female throughout her life is determined at her fetal stage. When she reaches the age of puberty then those primary oocytes undergo the process of meiosis phase 1 and a single cell divides into two and the number of chromosomes reduced to half. Now this is called as secondary oocyte. This is the pulla cell. As you know that ovary releases one egg every month. So it takes place alternatively. Now this secondary oocyte undergo the process of meiosis phase 2. This is called as ovary and follicle or graphy and follicle. It gets released on every 14th day of each month by the process of ovulation. Do you know what happens after it gets released? I will explain it while explaining fertilization. Now let's understand about egg. It is surrounded by two membranes, namely, outer membrane and inner membrane. It is filled with cytoplasm. It contains nucleus with 23 chromosomes. So that was all about a human female gamete. So that was all about gametogenesis in both male and female. Now let me give you a quick recap of whatever we have studied till now. Reproduction in humans consists of three phases: pre-fertilization, fertilization, and lastly post-fertilization. Till now we have covered two topics of pre-fertilization. Those are reproductive maturity and gametogenesis. The last topic of pre-fertilization phase is gamete transfer. Gamete can be transferred by two ways. First, by copulation. Second, by clinical methods. So that's all for this lecture. In the next lecture we will study about fertilization and what if fertilization does not take place.